Tonight, when you fall asleep, your brain will do something it has done every night for 1.8 million years. It will split your sleep into stages. It will cycle through them in a precise sequence. It will flush waste from your neural tissue, transfer memories from short-term to long-term storage, and regulate the hormones that govern almost every function in your body for the next 24 hours. Homo erectus emerged approximately 1.9 million years ago. They were the first members of the human lineage to look recognizably like us in body proportions. Taller than their predecessors, with longer legs built for endurance, a reduced gut, and a larger brain than any hominin that had come before. They lived in a world without walls, without roofs, without artificial light. Their night was genuinely dark. Their ground was genuinely hard. And their predators were genuinely active after sunset. The sleep architecture that Homo erectus developed under those conditions is, in every measurable way, the same architecture your brain runs tonight in your bedroom. That is not a metaphor. The staging, the cycling, the timing, the neurochemistry, all of it is identical. The only difference is that you are running a 1.8 million-year-old survival program inside conditions it was never built for. The architecture of sleep. What Homo erectus? Built your sleep does not switch on like a light and switch off when you wake. It moves through a repeating 90-minute cycle, typically 4 to 6 times per night. Research from Matthew Walker at UC Berkeley and published studies in the journal Nature Neuroscience identify 4 distinct stages. N1 is the entry point, a brief transition between waking and sleep. N2 is where your brain produces sleep spindles, short bursts of neural activity associated with memory consolidation. N3 is slow-wave deep sleep, where your brain emits delta waves and your glymphatic system opens to flush toxic metabolic waste from neural tissue. And REM, rapid eye movement sleep, is where memory transfer, emotional processing, and neural maintenance all converge. Each full cycle takes approximately 90 minutes. That number is not arbitrary. Research published in PNAS by Yuka Sasaki and colleagues at Brown University confirmed that the 90-minute ultradian rhythm maps to a fundamental oscillation in human brain activity that appears to be hardwired, not learned. What is particularly significant about the staging sequence is its order. Deep sleep is front-loaded in the night. REM is backloaded. If you cut your sleep by 90 minutes, you do not lose 90 minutes distributed equally across all stages. You lose almost all of your REM sleep because REM sleep occurs predominantly in the final 2 cycles of the night. The glymphatic system, your brain's nightly waste disposal. In 2013, a research team led by Macon Nedergaard at the University of Rochester published a study in Science that changed how neuroscientists think about sleep. They discovered a biological waste clearance system in the brain that is almost entirely inactive during waking hours and operates at full capacity only during deep sleep. They called it the glymphatic system. During N3 deep sleep, your brain cells shrink by approximately 60%. That shrinkage opens channels between neurons, and cerebrospinal fluid floods through those channels, physically sweeping metabolic waste products out of your neural tissue and draining them down to your lymphatic system for disposal. One of the primary waste products cleared by this system is amyloid beta, the protein fragment associated with Alzheimer's disease. Research published in Science by Shokri Kajori and colleagues confirmed that even a single night of sleep deprivation produces a measurable increase in amyloid beta accumulation in the human brain, particularly in the hippocampus and thalamus. Your brain is generating metabolic waste constantly, every hour you are awake. The glymphatic system is the only mechanism your body has to remove it at scale, and it will not run properly on broken sleep, short sleep, or sleep interrupted by artificial light. Homo erectus did not know any of this, but their sleep conditions, dark, cool, socially co-regulated, physically grounded, produced the precise environment this system requires, not by design, by survival pressure. REM sleep, memory, and the emotional brain. REM sleep is the most metabolically expensive phase of the sleep cycle. Your brain during REM is consuming almost as much glucose as it does when you are awake, yet your body is essentially paralyzed. The paralysis is intentional. During REM, your brainstem sends inhibitory signals to your motor neurons, preventing you from physically acting out the neural activity occurring in your cortex. Research by Robert Stickgold at Harvard Medical School, published in Nature Neuroscience, established that REM sleep is the primary window for procedural memory consolidation and emotional memory processing. During REM, your hippocampus replays the day's experiences and transfers the relevant information to your neocortex for long-term storage. Your amygdala processes the emotional charge of those memories. Research from Matthew Walker's lab, published in Current Biology, found that REM sleep specifically strips the emotional intensity from threatening memories while preserving the informational content. This is your brain running maintenance on the threat response system. It keeps the memory of the dangerous event, but reduces the alarm signal attached to it. What this means, practically, is that REM sleep is not rest. It is active surgery on your emotional and cognitive architecture. Chronic REM deprivation does not simply make you tired. Research published in the Journal of Neuroscience shows it produces heightened amygdala reactivity, impaired prefrontal regulation of emotion, and reduced capacity for accurate threat assessment. Every video on this channel goes this deep into the biology of what your body is actually doing and why. The people watching consistently report that understanding the origin of these systems changes how they treat them. If that kind of knowledge matters to you, this channel will keep delivering it. The next video is already waiting. It is 1.8 million years ago, East Africa, the dry season. You have been walking for 6 hours. Your legs are spent. Your feet carry the bruised print of volcanic rock and dry earth. The light is gone now, and the savanna sounds different in the dark. The insect frequency has changed. Somewhere in the mid-distance, a hyena calls. You lie down. The ground is hard. The air is cooling. Your core temperature has already begun to drop, and that drop is a signal, not a discomfort, a trigger. Your suprachiasmatic nucleus in the hypothalamus, your body's master circadian clock, is reading the falling temperature and the absence of blue light. It is releasing melatonin. You don't fight it. You have never fought it. Darkness means sleep, and sleep means survival. Within 8 minutes, you are in N1. Your thalamic gating system has partially closed the sensory relay between your body and your cortex. Sound is still reaching you, processed lower in the brain, but the volume has been turned down. You are not unconscious. You are in an in-between state. Your body evolved specifically for an environment where complete unconsciousness would have been lethal. 20 minutes later, you are in N2. Your brain is producing sleep spindles, 12 to 15 Hz bursts of thalamocortical activity. Research by Jan Born at the University of Tübingen, published in Nature Neuroscience, confirmed that these spindles are the mechanism by which motor and procedural memories from the day are locked into cortical storage. What you learned today, which plants were safe, which terrain was unstable, which sounds preceded predator movement, is being filed permanently. 40 minutes in, you enter N3. Your delta waves are now dominating. Your glymphatic channels have opened. The amyloid beta, the tau fragments, the metabolic debris from a day of high-output cognitive work, navigation, threat assessment, tool use, social calculation is being cleared. Your growth hormone is peaking. Your immune system is rebuilding. A wound on your forearm from 3 days ago is laying down new collagen tonight at a rate 4 times higher than it would during daylight hours. 90 minutes after you closed your eyes, something shifts. Your brainstem sends a different signal. Your motor neurons are inhibited. Your limbs paralyzed by your own nervous system. But your brain is surging. You are in REM. Your eyes are moving beneath closed lids. You are processing the near miss from yesterday when a predator passed within 30 meters of the group. Your amygdala is replaying it. Your hippocampus is tagging the spatial coordinates. Your prefrontal cortex is running simulations of alternative outcomes. When you wake, you will not feel the fear of that event as sharply, but you will remember exactly where it happened and what preceded it. This cycle will repeat 4 more times before dawn. Each time, the deep sleep stage shortens slightly, and the REM stage lengthens. By the 5th cycle, your REM window is 45 minutes long. Your emotional archive for the day has been processed, indexed, and stored. You wake at first light. Not because of an alarm, because your cortisol level has been rising since 4 a.m., a programmed physiological dawn signal your adrenal glands have been executing since before Homo erectus existed. Your brain is running the exact same program that Homo erectus ran 1.8 million years ago. The program has not changed. The environment has. The single biggest change is light after dark. Your suprachiasmatic nucleus uses blue wavelength light as its primary signal for daytime. Smartphones, LED screens, and overhead lighting all emit blue dominant light. Research by Charles Czeisler at Harvard Medical School, published in the Proceedings of the National Academy of Sciences, found that evening blue light exposure delays melatonin onset by up to 90 minutes and reduces REM sleep duration in the subsequent night. Your core body temperature needs to drop approximately 1 degree Celsius to initiate sleep onset efficiently. Research published in Current Biology by Natalie Ranjbar and colleagues confirmed that bedroom temperatures above 18 to 20 degrees Celsius measurably reduce slow-wave, deep sleep duration. Homo erectus slept in open air where overnight temperatures in East Africa typically dropped to between 15 and 20 degrees Celsius. Your body is still calibrated for that range. The third factor is sleep timing. Your circadian rhythm was calibrated around a solar cycle in a specific latitude range. Research from Kenneth Wright at the University of Colorado, published in Current Biology, found that camping outdoors for 1 week without artificial light shifted participants' melatonin onset timing by 2 hours earlier, aligning with natural sunset. Their cortisol awakening response also synchronized more tightly with sunrise. What this suggests, and the research is consistent here, is that the primary enemies of the sleep program your brain is trying to run are not physical. They are environmental mismatches. Light at the wrong time, temperature too high. Sleep timing shifted 2 to 3 hours later than your biology expects. None of these mismatches are dramatic. None of them feel catastrophic in the moment, but they are accumulating. Every night you run the program in a degraded environment, you are taking a withdrawal from a biological account that Homo erectus spent 1.8 million years building. Tonight, your brain will begin its first 90-minute cycle, approximately 15 minutes after you close your eyes. Your thalamus will reduce sensory input. Your glymphatic system will open. Your hippocampus will begin transferring today's experience into long-term storage. Your amygdala will process every emotional event since you woke this morning. None of that requires your permission. Homo erectus built this program under selection pressure so extreme that any individual whose brain did not run it reliably did not survive long enough to pass the variation on. You are not fighting poor sleep habits. You are running a 1.8 million year old survival program in an environment it was never designed for. Understanding that distinction is the beginning of working with your biology rather than against it. The next video on this channel looks at something your gut has been doing since before Homo erectus existed. A biological event that happened 1 million years ago changed the size of your digestive tract, the volume of your brain, and the bacteria still living inside you right now. That one is coming next, and it concerns every meal you have eaten today. This channel exists for people who want to understand what their body is actually doing and why. That understanding belongs to you.